Hello student, good afternoon. Today we will start the next topic, that is the larval forms of trematoda. In last period we have to completed the two larval forms, that is Meracidium and Sporocyst, and remaining two larval forms we will complete it in this lecture. Third stage, first stage is Meracidium, second is Sporocyst, and third stage is Redia. Means today we will start the two larval forms, the Redia larva and Cercaria larva and Metacercaria. Redia larva. The Redi emergence from the Sporocyst, means Redia are developed in the body of Sporocyst larva by the rupture of body wall of the Sporocyst. Each Redia is an elongated and cylindrical form measuring 1.3 to 1.6 millimeter in length. It has a circular ridge called collar near the anterior end and a pair of projection termed lapetus near the posterior end. The body wall consists of the usual cuticle, musculature and subepithelium. Unlike the Sporocyst, each Redia possesses a mouth located at the anterior terminal of the elongated body. The mouth leads into a musculature pharynx, which in the turn leads a blind sac. The gut, numerous unicellular pharyngeal glands open into the pharynx. Protonipridia divided further and form a much branched system. All the flame cells of one side open out through a common excretory ducts. Birth pore is present lateral to and in proximity of the mouth. Within the brood chamber are found in germ balls. Redia is a very active little creature. It moves about the contractions of the body and lapates. It nourishes itself by sucking fluids and cells of the host tissues. With its muscular pharynx, it soon migrates into the digestive glands of the snail during the summer months, when the food is sufficiently available. Mother Redi mature in 12 to 18 days. The germ balls give rise to the second generation of daughter Redi. Germ balls of daughter Redi in the winter develop into the next larval stage, the Cercaria. That is the Redia stage. At the anterior side, mouth, below the mouth, pharyngeal glands, pharynx, muscular collar, birth pore also near the muscular collar, gut, protonipridia or flame cells, daughter Cercaria larvae, oped or promocrustacum, cuticle, outer surface of the body, that is the cuticle, and germ cells, inner cell layer, that is called as epithelial cells. This is the Redia, morphological structure of Redia. Next larva, Cercaria larva. The Cercaria leave the mother or ready through the birth pores from each redia. 14 to 20 Cercaria are produced. Means each and every redia to produce the 14 to 20 redia in the body of redia. That is the Cercaria, to produce the Cercaria in the body of redia. Cercaria is about 0.25 to 0.35 millimeter long and is an oval larva with a tail. The body wall consists of cuticle, musculature and subepithelium. On the anterior side, there is a mouth with a sucker or suctorial pharynx, long esophagus and a brief intestine. Number of dark brown cells called cystogenous glands are found beneath the cuticle. There is a pair of excretory ducts, each with several phlegm cells. These open near the hind end of the larva into the bladder that leads out through a pair of nephridopores. A nervous system similar to that of adults has also appeared. Rudiments of reproductive organs develops the germ cells near the hind end. That is the morphological structure of Cercaria. At the anterior side, that is oral sucker. Below the oral sucker, musculature pharynx, esophagus, gut cecum, cystogenous gland cells, both the lateral side of this Cercaria, phlegm cells, also both the lateral side, acetabulum at the ventral side. Germ cells also present, excretory vesicle, excretory ducts and tail also. Tail is the swimming organ, swimming from the one place to another place. Use of tail, and enters in the body of definitive host. That is the Cercaria larva. Last and infective stage of larval stage of trematoda affect to the any vertebrate animals, human and other animals also. From the digestive glands of the snail, the Cercaria pass into the pulmonary sac and the escape into the surrounding water. Means all these larval forms develop in the body of snail. Snail is the intermediate host, and after Cercaria larva develop and escape out from the body of snail. Means Cercaria larva is exit out from the body of snail and to search the next definitive host, that is man and other vertebrate animals. Pulmonary sac and their escape into the surrounding water. Cercaria swims about 5 minutes to an hour in summer. Afterwards, it settles down on the some green leaves of water plants. Tail is cast off and the body becomes rounded and forms around itself a thick brownish cyst wall from the cystogenous glands. The encysted Cercaria is now called as metacercaria. And that Cercaria is not any chance to enters the body of vertebrate host. At that time, insisted larva, that Cercaria larva attach on the any part of vegetables or the leaves and insisted cyst wall is created, and that larva is called as metacercaria. And that type of metacercaria, that at the time of drinking of waters, some animals, that time contamination of water along with metacercaria enters in the body of that definitive host, man and other vertebrate host, and it ruptures the cyst wall and it develops into adult worm in the body of definitive host. Okay. Factors affecting development in the snail. Factors affecting development in the snail. These factors affecting development in the snail. The rate of development in the snail host in a function of the environmental temperature. Kendall reported curious effects of the temperature on the development of fasciola. In snails, maintenance of the laboratory temperature, Dr.D.D. were not produced the first generation of Redia, giving rise to Cercaria. On the other hand, snail exposed daily to a period low temperatures, that is 4 to 5 centigrade, daughters Redia were produced from the Redia. The state of snail nutrition also affect development. The effect of the food availability on the development in the snail host is probably the explanation for the observation that fewer larvae develop in small snail than in the large ones. Metacercaria, last larval stage. It is juvenile flukes having a rounded form with a diameter of about 0.2 millimeter. It resembles Cercaria larva but differs from the latter in the absence of the tail, cystogenous glands, and presence of the thick cyst wall. Excretory bladder opens directly through the single pore at the anterior side, that is mouth, cyst wall on the surface of that body, oral sucker also at the oral region, musculature, esophagus, intestine, excretory canals also, both the lateral sides, phlegm cells also present, excretory vesicle and acetabulum, oral sucker and acetabulum. That is the metacercaria. The metacercaria insisted on the vegetation, may live for as long as a year at low temperature, or for a short time as 2 or 3 weeks at a 25 degrees centigrade. They cannot be die. Infection of final host. The final insisted in the host requires an elevated temperature, that is 38 degrees centigrade, redox potential, a high carbon dioxide tension, and the presence of bile. The factors necessary for the existence of fasciola metacercaria seems to serve as the stimuli to activate the worms, but the mechanism is not clearly understood. When the metacercaria reaches the intestine of host, the cyst is digested and young flux is liberated, the young worm penetrates the wall of gut and passes into the peritoneal cavity. In the body cavity, the young worms seems to the move about rather aimlessly until they encounter the capsule of liver. On contact with the liver, the flukes burrowing into the developing liver parenchyma. As the move, they grow in the size within the 5 to 6 weeks and finally settle in the bile duct. Each attains sexual maturity and begins to lay eggs 11 to 13 after ingestion by the mammalian host. This is the two larval stages, Redia and Cercaria. We have to complete it. Okay, thank you.